Good afternoon. Welcome to MSK Science Spotlight. My name is Danwei Huangfu. I am an associate member in the developmental biology program at the Sloan Kettering Institute. This seminar series features today's leaders in basic and translational biomedical science. If you have questions throughout the talk, you can email them to the Office of Scientific Education and Training at OSET, O-S-E-T, at mskcc.org, or by tweeting at MSK Education. All questions will be answered at the end of the program. For the best viewing experience, please use Google Chrome. If you're using Internet Explorer, please use version 11 or higher. If you're experiencing technical issues during the live stream, please lower the video resolution. You can do that by clicking on the gear shift on the lower right of the screen. Now, let me get to the fun part. It's my great pleasure introducing today's speaker, molecular biologist Scott Keeney, a member of the molecular biology program here at the Sloan Kettering Institute, Howard Hughes Medical Institute investigator, and Frederick, our Adler chair. Dr. Scott Keeney's lab investigate the mechanisms that controls the formation of DNA double-strand breaks during meiotic recombination. He is well known for his work on SPO11, which generates DNA double-strand breaks that initiate meiotic recombination. In fact, the timing couldn't be better, as Scott were just elected to the National Academy of Sciences this week, together with Chris Lima, also from our institute. So congratulations, Scott. His talk today is entitled, How Do Little Chromosomes Know That They Are Little? Scott.
Thanks, Dominic. I want to thank MSK for giving me the opportunity to talk about some of the research in the lab. It's a real pleasure to be able to address all of you. I hope you're all staying safe. Please do send your questions in. I'd be happy to answer them at the end. So as you all probably are aware, every one of us went through a pair of extreme genetic bottlenecks when cells in our parents reduced their diploidy content to haploidy in order to generate sperm or eggs for sexual reproduction. And the specialized cell division that reduces the chromosome copy number from 2N to 1N is, of course, the cell division we call meiosis. And that's the key step in germline development that provides for this step in sexual reproduction where haploidy is reduced, genome ploidy is reduced so that it can then be restored when the egg is fertilized by the sperm. My lab is interested in understanding how meiosis works and how the chromosomes are segregated to reduce the genome complement. So before I talk about meiosis, though, I just want to orient people by contrasting it with the cell division of mitosis. So what's cartooned here is a very simple diploid organism with a pair of homologous chromosomes. You can imagine it got one chromosome from its mom and the other chromosome from its dad. And the goal of mitosis is to carry out a faithful chromosome segregation so that the daughter cells have the same genome complement as the starting cell. And cells do that by doing one round of DNA replication that generates a pair of sister chromatids for each homolog. And then the sister chromatids are pulled apart from each other by the spindle. And then after cytokinesis, you end up with daughter cells that have the same genome complement as their mother cell. So there's a fundamental logic by which all spindles in eukaryotic cells work. And that logic is that the DNA molecules that you want to separate first have to be connected. In mitosis, these connections come predominantly in the form of what we call sister chromatid cohesion, which is protein complexes that you can think of as acting like glue that hold the sister chromatids together. That glue then resists pulling forces from the spindle microtubules, and that allows the cell to know that the chromosomes are attached properly to the spindle. And then the glue can be dissolved to allow the sister chromatids to separate from one another at the first division. The goal of meiosis is different. As I said, the goal of meiosis is to reduce the genome complement. And so what cells have to do here is to do one round of DNA replication, but now two successive rounds of chromosome segregation. Now, the way this is cartooned, the second meiotic division is overlaid with the mitotic division, and that's because meiosis 2 is a lot like mitosis because it separates the sister kinetochores from each other. Meiosis 1 is the one that's unique, though, because that separates the homologous chromosomes from each other. So now we have a problem, though, because again, the spindle logic is that the molecules you want to separate first have to be connected. And in mitosis, the sister chromatids, at least simplistically, you can think about how the cell solves the problem of identifying what DNA molecules are sisters, because the sister chromatids are born in spatial proximity to each other when the strands of the parental duplex are split apart and copied. So the cell can at least, conceptually, at least it's easy to understand how the cell knows that these two DNA duplexes were the sister chromatids of each other. But the homologous chromosomes need not have had any kind of special relationship with each other. In a simple diploid, it's not a big deal. The red and the blue just need to pair with each other. But most real diploid organisms have many more pairs from that. And as you'll see, in budding yeast, there are 16 pairs. We have 23, mice have 20. So this is a much more challenging problem for most organisms. So the way this works is that the cell lines up its homologous chromosomes next to one another, and they exchange some genetic information in a process I'll tell you about in a second. And that then forms these physical connections between the homologous chromosomes that allow them to then solve the spindle logic. These connections come in the form of what we call crossover recombination products. So you can see here that this red chromatid has crossed over and physically become joined to the blue chromatid here. And I hope you can appreciate that the spindle microtubules now here emanating from the top might connect to the pair of sister centromeres here. And then that pair of sister centromeres is contiguous with this blue chromatid arm, which swaps over to the red one, which is held by sister chromatic cohesion to its sister, which is contiguous with the other pair of sister kinetochores that attaches to the opposite pole. And perhaps you can see how this whole ensemble then would come under tension when the spindle microtubules start to pull. And that tension is what tells the cell that this chromosome pair has connected properly to the spindle. Then dissolving the sister chromatic cohesion on the arms would allow the homologous chromosomes to separate from one another. So the name of the game in meiosis is identifying these homologs and lining them up along their lengths. And that's the process I want to talk about. So we'll just focus on these very early events right after DNA replication as the chromosomes pair with one another. What happens is that in most organisms, the cell accomplishes the task of figuring out which DNA molecules are homologs by doing this kind of strange process where it deliberately damages its own DNA at multiple places. Then in order to repair that DNA, the broken ends have to search for a homologous DNA duplex to serve as a template for repair. That search allows the homologous chromosomes to find one another, line up along their lengths, and then one or a small number of the recombination reactions ends up as that crossover that I told you about is necessary to stabilize the pair at metaphase 1. So most of what we know about how this process works comes from studies in a number of model organisms, including budding yeast, which is what I'll talk about today. What's cartooned here now is the two strands of a DNA duplex. So you have to imagine there's a sister chromatid in red that I'm not showing you. This DNA damage that's cartooned here as these green stars is actually double strand breaks in the DNA that are made by this protein SPO11. SPO11 is a topoisomerase relative. And so like topos, it cuts DNA through a covalent protein DNA intermediate. Then in order for that break to be repaired, SPO11 has to be removed from the ends so that the ends can be processed and repaired. And what happens then is the cell nicks the DNA strands that SPO11 is attached to. That releases SPO11 attached to these short oligonucleotides and frees up the ends for further processing. That gives you single-stranded DNA that can now search for a homologous DNA duplex and invade it. Then the cell has a very highly regulated set of reactions that completes the repair process. And one possibility is that the broken DNA can copy a little bit of information from its attacked partner and give this patch-like repair we call a non-crossover. Or the cell can build these branched DNA structures called double holiday junctions that most of the time get resolved as crossovers. So one of the interesting things to me about this process is that this is actually kind of a crazy thing for the cell to do, because double strand breaks are known to be a highly toxic form of DNA damage. And yet the cell makes lots of these double-strand breaks in order to pair up and recombine its chromosomes. And to give you a flavor for how frequent that really is, this is a mouse spermatocyte that's been spread on a microscope slide and stained for a chromosome structure protein called SYCP3 and for a repair protein called RAD51. That's one of the proteins that carries out this search for homology. So each of these green spots on this chromosome spread is a place where there's a double-strand break that's in the process of being repaired. And I hope you can appreciate there are a lot of these spots. So if you count them up, we come up with an estimate that each mouse meiotic cell makes on the order of 200 to 300 double-strand breaks per meiosis. If you made that many double-strand breaks in a somatic mouse cell, you'd be pretty much guaranteed to kill it. But every meiotic cell does this as part of its normal development, which I think is really interesting. So as it turns out, errors in this process are a leading cause of miscarriage and congenital birth defects in humans. And so the basic point here is that if the cell doesn't do this properly, it can't segregate its chromosomes. And so you have to have these double-strand breaks in order to have successful sexual reproduction. But these breaks, as I said, are a potentially lethal form of DNA damage. And so the cell needs to kind of balance these positive and negative aspects of this very interesting process. So my lab's been interested in this for a long time. We're interested in understanding how double-strand breaks are formed. We're interested in how cells control the number, timing, and location of the breaks. And we're interested in how cells control the repair process that deals with the breaks once they're formed. And most of what we do is studies in either mouse or in budding yeast. And the story I'll tell you today is a budding yeast story. So this story mostly deals with the mechanism of break formation and the mechanisms that control when, where, and how many breaks there are. So all of this work that I'm going to tell you about was led by a really talented research scientist in the lab, Hajime Murakami. And you can read about this in this preprint that's on bioRxiv, and it's soon to be out in peer-reviewed form. Okay, so back to this cartoon of the necessity of making these DNA damage events on the chromosomes and then forming at least one of these crossovers. So one of the key points here is that every pair of homologous chromosomes has to have at least one crossover in order for them to segregate properly. Therefore, you have to have at least one break. You can't have a crossover if you didn't make a break. So we can kind of push the problem back to that stage and say, well, how do cells make sure that every chromosome pair gets at least one break? So as I mentioned before, Saccharomyces cerevisiae has 16 chromosome pairs, and they range really dramatically in size. So this is a scale cartoon that the smallest chromosomes range from about 200 to 300 kilobases, and the longest are over a megabase. And so there's a really wide variety of sizes of the chromosomes. And it turns out that yeast makes about 150 to 200 double-strand breaks per meiosis. So if you were to distribute those breaks randomly across the genome, the question that Hajime wanted to know is, would randomly distributing that many breaks be enough to be sure that you always got at least one break on all of the chromosomes, particularly the little ones? And the answer was no, it didn't always work. So he did simulations where he randomly distributed 200 double-strand breaks across the yeast chromosomes. And he found that in the random simulations, he got about a 3% failure rate. So that meant about 3% of meioses had at least one chromosome pair that didn't get a break at all. So that may not sound like a lot, but in evolutionary terms, if you have a 3% hit on your reproductive fitness, that's a pretty big deal. So what was more interesting was that if he looked at which chromosomes were the ones who were most likely failing to get a break, as you might expect, it was the littlest chromosomes. So the smallest chromosomes in yeast are chromosomes 1, 6, and 3. And you can see that if you range out the failure rate, what percent of meioses fail to get at least one break on the chromosome, and plot it out as a function of chromosome size, you can see the little chromosomes are really the ones that are most at risk. So how do cells mitigate this risk of failing to make a break on the little chromosomes? Well, it turns out that breaks are not randomly distributed across the genome. And in fact, the little chromosomes make a lot more double-strand breaks per kilobase of DNA than larger chromosomes do. So what's plotted here is a map of where the double-strand breaks are, and we do that by sequencing those little oligonucleotides that I told you were covalently attached to SPO11. those oligos tell us where SPO11 has made double-strand breaks. And if we simply add up those number of sequencing reads from these SPO11 oligos and plot them as a function of chromosome size, where we're looking at the density of these oligo reads, so reads per million per kilobase of DNA, you can see that most of the chromosomes have this particular density of reads per kilobase of DNA, but the short chromosomes really are making a lot more double-strand breaks than the little ones. Now, in total number of double-strand breaks, the big chromosomes make more, but per kilobase, the little chromosomes are a lot hotter. So how does that happen? So some really beautiful work from Franz Klein's lab at the University of Vienna a number of years ago had shown us that short chromosomes also accumulate a lot more of this protein REC114. Also some other proteins, but for the simplicity in this talk, I'm just going to focus on REC114. So what is REC114? Turns out that's a partner of SPO11 that's absolutely essential for SPO11 to cut DNA. So the idea here is that perhaps the way cells are controlling this is the little chromosomes are better at recruiting the proteins that are necessary to make double-strand breaks, including REC114, as partner of SPO11. And because they recruit more of the double-strand breaks, they're able to make more double-strand breaks per kilobase than their bigger brothers. So, but then that just kind of pushes the problem back one more step. How do the short chromosomes recruit break proteins at a higher density? How do the little chromosomes know that they're little and compete more effectively for binding to these proteins? So that's the question that Hajime really set out to do in this project. So one of the experiments that he did was to do a detailed time course across meiosis. So you can take budding yeast and induce a culture of budding yeast to enter meiosis and proceed through fairly synchronously. And if you collect time points at different times after you've induced meiosis and measure the binding of REC114 across the genome by ChIP-seq, what you can see is that of course, at the beginning of meiosis, there's no REC114 on the chromosomes, and that makes sense because I didn't tell you, but REC114 is a meiosis-specific protein, so it's not even expressed at this time point. But the protein begins to be expressed, and at very early time points, you begin to see accumulation of the protein on the DNA, and then it accumulates to higher levels, and then it begins to go away again. Double-strand breaks start being made at around 2 hours, and they continue to accumulate until roughly about 4 hours. So this is the period of time when double-strand breaks are being made, but they first start being made when the protein levels are still quite low. So what's interesting is if you look at how this protein is distributed across the chromosomes according to their size, you see that the little chromosomes have more of the protein throughout meiosis, early, middle, and late, but the pattern is a little bit different on all the chromosomes as a whole. At the very earliest time points, which again is when the protein just starts to be synthesized and is just binding to the chromosomes, you can see the little chromosomes have a lot more of the protein than the bigger chromosomes do. And the bigger chromosomes really don't show much of a size relationship at all. Basically, all of them recruit REC114 at about the same density. At the peak time, when there's lots of REC114 bound to the chromosomes, the little chromosomes still have more than the others, but now there's more of a linear relationship across all of the chromosomes, and that linear relationship persists or even maybe gets a little stronger as the protein is disappearing from the chromosomes. So Hajime set out to try to understand this, and one of the things that he did was to measure in detail the timing that REC114 binds to and then dissociates from the chromosomes. And so what you're looking at is the smallest chromosome, chromosome 1, and the largest chromosome, chromosome 4. And each of these little red dots is a peak in the REC114 ChIP-seq profile. And for that peak, he measured the time of association of REC114 to that site. And so you can see lots of association times, and you can see the vertical line here is the average for that chromosome. And I hope you can appreciate that the average for the chromosome, even though there's a lot of variation, the average is a little bit later than the average for the first chromosome, for the smallest chromosome. In other words, the longest chromosome is accumulating REC114 a bit later than the shortest chromosome is. He did the same to measure the dissociation time, and you can appreciate, I hope, that the little chromosome is dissociating REC114 from it a little bit later than the longer chromosome. So then, of course, you can just measure the time difference between association and dissociation and get the duration time at that position. So when Hajime looked at all of the chromosomes, this is what he saw. So now chromosome size is on the vertical axis and time in meiosis is on the horizontal axis. And what you can see is that the small chromosomes start to accumulate REC114 earlier than the bigger chromosomes do. And the bigger chromosomes really don't have much of a size relationship. It's just sort of a scatter of points here. But at the dissociation time, it's different. The little chromosomes, again, are a bit later on average, but now there's a nice linear relationship that relates most of the chromosome dissociation times from each other. And the net result of these two timings is that the duration is negatively correlated with chromosome size. So REC114 spends longer bound to the little chromosomes than it does to the longer chromosomes. And we think that's probably a big part of how the little chromosomes are able to make more breaks, because they spend more time associated with the proteins that make double-strand breaks. All right, that's fine, but how does that work? How do the little chromosomes know that they're little? So to summarize what I just showed you in this cartoon form, you can see that the long chromosomes tend to accumulate REC114 a bit later than the small chromosomes, and they dissociate REC114 from them a bit earlier than the small chromosomes do. So we can focus on both association time and dissociation time. And first, I want to talk about this dissociation time, when REC114 comes off, because we actually already understood a big part of how this works. And that was from previous work from a graduate student in the lab, Drew Thacker, that I'll summarize in a second. But I also want to draw your attention to this really lovely paper that was published last year from Andreas Hochwagen's lab at NYU. And this was work of Vijay Subramanian, who has her own lab in India now. So what Drew found was that when homologous chromosomes find one another, a process that we call homolog engagement, that sends some kind of signal that inhibits further double-strand break formation. So this is a form of negative feedback where once the homologous chromosomes have succeeded in pairing with one another, they don't need to make breaks anymore. And so in fact, they stop making breaks. So that homolog engagement is dependent on many factors, but the one I'll just mention for the purpose of this talk is this protein ZIP3, which is important for the cell carrying out regulated repair of these double-strand breaks to make crossovers. So it's not important for our purposes to know exactly what ZIP3 does. What you just need to know is that if you don't have ZIP3, the cell is defective for homolog engagement, and so it does not shut down break formation properly in late stages of meiosis. And as a result, even after the chromosomes align with each other, they keep making double-strand breaks when they should have stopped. And so I showed you this graph before. This is, again, binding of REC114 chip signal, binding of REC114 to the chromosomes arrayed according to their chromosome size. And you can see at the early time point, smallest chromosomes have this pattern I pointed out before, where they have very high levels of REC114. In a ZIP3 mutant, you get exactly the same result. And that makes sense because ZIP3 is something that's acting later to control late processes once the chromosomes have actually already engaged one another. So this early time when the proteins are just starting to assemble and the homologous chromosomes haven't started to engage one another, ZIP3 status is irrelevant. But if you look at the later time point, when there's this nice negative correlation between chip density for REC114 and chromosome size and wild type, that correlation is completely gone in the ZIP3 mutant. So ZIP3 is controlling the size dependence for persistence of REC114 on the chromosomes. And in a ZIP3 mutant, one way to look at this is that the small chromosomes don't know they're little anymore. The small chromosomes are not special for retaining REC114. So that and a bunch of other evidence that I don't have time to go into has led us to conclude that this double-strand break feedback control through homolog engagement is a big part of what controls this differential timing of REC114 dissociation from chromosomes. Okay, that's fine, but then what controls the differential association timing? It turns out that there's many factors that contribute to that. And in the interest of time, I can't go through each of these in detail. But what I can tell you is that replication timing is important. What Hajime showed in a previous paper is that replication timing is coupled to the timing of REC114. And in particular, what he found is that early replicating regions accumulate REC114 early. So an interesting possibility could have been that association time was simply reflecting that little chromosomes tend to replicate earlier than bigger chromosomes, which they indeed do. And so it turns out that is part of how the association time of Rec114 is controlled in meiosis, but it's only part of it. It doesn't explain everything. Hajime also found in this study that the current study I'm talking about now, that proximity to centromeres and telomeres also governs the timing of Rec114 association. So he asked a simple question. He asked, if I just take these three factors that I know control the association time for Rec114, can I build a simple model that explains the association time as the combination of those three factors? So he did a multiple regression analysis and asked whether he could predict Rec114 association time as a function of the replication timing of a section of chromosome, its proximity to the centromere, and its proximity to the telomere. And so what you're looking at here is a graph where each of the points is a binned part of the genome, about 20 kb, I believe, and where he's built this regression model and simply predicted the time at which Rec114 would associate with the chromosome and compared it to when it actually associates with that segment of the chromosome. And what you can see is this regression model actually does a pretty good job. It explains about half of the variance in the time of Rec114 association. And so that means that just these three factors, replication time, centromere proximity, and telomere proximity, are sufficient to explain quite a bit of the variation. But what I'm showing you here is for the larger chromosomes. If you look at bins on the small chromosomes, you get a different answer. The short chromosomes are not actually well predicted by this model. And in particular, what you can see is most of them are in this part of the graph, which means that their predicted time, their observed time is much earlier than theirs predicted by the model. In other words, this model does a good job of explaining things through most of the genome, but it doesn't do a good job on the little chromosomes. And the short chromosomes are actually doing a better job of recruiting Rec114 early. So this led Hajime to hypothesize that there's something intrinsic to the sequence of the small chromosomes that boosts Rec114 accumulation on them, specifically at these early times. So if that's true, then you can make some simple predictions. One of those predictions is that if you change the chromosome size for the little chromosomes by fusing them to a larger chromosome, they should still know that they used to be small, and they should still act like a small chromosome. Because what we're hypothesizing is that there's some kind of intrinsic sequence feature that boosts Rec114 association. So Hajime did this experiment. He took chromosome 1, which is the smallest chromosome, and he bisected it and fused it to chromosome 4, the largest chromosome. And so he gets two derivative chromosomes that are on either intermediate or long for budding yeast. And then he did the ChIP-seq experiment again and asked how Rec114 binds to these chromosomes. So what you're looking at here is, again, this plot of Rec114 ChIP-seq density as a function of chromosome size at a very early time point. So this is right when the hypothesized boost is happening. And you can see that all of these time points for the, points for the long chromosomes are very similar to each other in Rec114 chip density. And there's no real relationship with chromosome size, but at this early time point, the little chromosomes really stand out. So this is a combination of both wild type and the translocation strain for everything except the translocation chromosomes. So here's where chromosome 1 left arm is in the wild type when it's a short chromosome. And so the prediction is that if it's going to now not realize that it used to be a little chromosome, if it's going to think it's a big chromosome, it should now have a chip density that's predicted by its chromosome size. But if it retains this hypothetical boost, then it should now, even though it's a big chromosome, it should act like a little chromosome and recruit lots of Rec114. And Hajime got a really beautiful result that was exactly as predicted by his model, which is that the translocated chromosome is still behaving like it's a little chromosome. And you can see that if you look at the density, chip density maps across the chromosome. So on the top is wild type. Here's chromosome 1. You can see there's a very high Rec114 chip density, and chromosome 4 has a much lower Rec114 chip density. And in the context of the translocation chromosomes, you can see that the derivative parts that came from the little chromosome still have a higher chip density. And you can see there's really dramatic switch from where the small chromosome sequences are to the long chromosome sequences. So this seemed to indicate then that there were at least these 4 factors that I mentioned before: replication timing, proximity to centromeres and telomeres, plus this special property of the little chromosomes that boosts those chromosomes specifically. So one implication of that idea is that, remember I told you back at the beginning, we got into this by thinking about what happened if you randomly distributed breaks across the genome, and realized that if you did that, the little chromosomes would be the ones that are at risk. But what's interesting, or among other things that's interesting about this, is that Saccharomyces species have kept 3 short chromosomes for a really long time. So what you're looking at here is a phylogeny of different species in the Saccharomyces clade. And here's Saccharomyces cerevisiae up here. But even this species that's 15 million years or so diverged from Saccharomyces cerevisiae still has the same karyotype as Cerevisiae. So this karyotype of 16 chromosomes with 3 little ones has been evolutionarily persistent for millions and millions of years. But we show that creates this situation where it has to compensate for having little chromosomes by having special mechanisms to boost the REC114 binding and boost double-strand break formation on them in order to mitigate the risk of chromosome missegregation. So what Hajime wanted to ask was, well, what would happen if you were able to do this evolutionary experiment where instead of having a short chromosome, you had a longer chromosome? Would you lose the boost? Because if the boost is there to help make sure that the short chromosomes segregate, you would imagine that once you're no longer at risk of missegregation, you might lose the selective pressure to maintain the boost. It turns out there was a natural experiment that Hajime could do to test this idea, because one of these species, S. macate, has acquired a translocation in between its last common ancestor and with these other species. And so that's cartooned here. Saccharomyces cerevisiae, chromosome 6 is the second smallest chromosome at 270 kilobases. Chromosome 7 is a little bit over 1 megabase. In S. macate, the ancestral version of chromosome 6 has been fused to the ancestral version of chromosome 7 to give these 2 derivative chromosomes that are intermediate in size. So the experiment is now to ask, in S. macate, where the chromosome 6 segments are no longer in the context of a small chromosome, do they behave like chromosome 6 does in Cerevisiae and have this boost? Or do they behave like a long chromosome because they've been in a long chromosome now for millions of years? So Hajime did ChIP-seq in S. macate and compared it to the ChIP-seq in Cerevisiae. And so the triangles here are S. macate ChIP-seq and the circles are S. cerevisiae ChIP-seq. And you can see that overall across the genome, the 2 species are very similar, indicating that these patterns I've been talking about are highly conserved evolutionarily. The longer chromosomes have a lower chip density, and the bigger chromosomes have a higher the smaller chromosomes have a higher chip density. So here's where Saccharomyces chromosome 6 left arm is. That's the blue arm here in the cartoon. It has a high chip density. But here's where the syntenic region of S. macate chromosome 6 is. So this is now, again, remember, in a long chromosome context. And so having removed the need for a high double-strand break frequency, what we surmise here is that that has relieved the selective pressure to maintain this high level of binding of REC114. So what I hope I've convinced you of is that the cell has these many complicated ways to apportion REC114 protein amongst the chromosomes. And it does so in order to mitigate chromosome segregation risks. So the double-strand break proteins, when they first start to be synthesized at the beginning of meiosis, bind preferentially to the little chromosomes through a variety of mechanisms. One is the fact that the little chromosomes tend to replicate a bit earlier. Another is that sequences close to telomeres actually tend to inhibit REC114 binding, but sequences close to centromeres activate REC114 binding through mechanisms we don't fully understand. And in addition, the little chromosomes have this boost that the bigger chromosomes don't have, and that boost really amps up the binding of REC114 to the little chromosomes. And so as a result, the little chromosomes spend longer in this double-strand break competent stage when they can make double-strand breaks and they can do recombination to compare their chromosomes. They can make this structure I didn't have time to talk about called the synaptonemal complex. And then once the chromosomes start doing that, they start to dissociate REC114 from them. And because the bigger chromosomes make more total number of breaks, it's lower density, but more total number, they pair sooner with one another, and so they tend to eject REC114 sooner through that homolog engagement pathway that I mentioned. Whereas the little chromosomes often take longer to find their partners, and so they undergo homolog engagement later, and that means they're slower to get rid of REC114, so they can stay in this double-strand break competent stage for a longer period of time. And as I showed you, this is playing out on evolutionary timescales as well, because the cell has to maintain this selective binding of REC114 in order to mitigate the risk of chromosome missegregation. So of course, this opens up lots of other interesting questions that we don't know the answers to yet, but Hajime is actively working on. One is, what is the nature of this boost? What is it about the little chromosomes that makes them really good binding targets for REC114 and MI4 and the other proteins that are essential for double-strand break formation? We don't know the answer to that. We don't think it's strictly an epigenetic phenomenon because we can propagate those translocated chromosomes for many generations, both mitotically and mitotically. And in Saccharomyces cerevisiae, they retain the boosting behavior even over many, many, many generations. So we think that a purely epigenetic mechanism is probably not the explanation. So we think there's probably some sequence elements or some other features of the chromosome that make them really attractive binding targets for REC114, and we're trying to figure that out. Another question is how does REC114 end up getting kicked off of the chromosomes when the homologs engage one another? We're very interested in understanding how that works. Finally, I want to close by saying that this is turning out to be, at least in its broad outlines, an evolutionarily recurrent theme, because other species seem to have independently acquired the ability to bind high levels of REC114 and its partners as a way to mitigate the risk of chromosome missegregation from having very small chromosomes. So in mouse and other mammalian species, the X and Y chromosomes in spermatocytes share only a tiny piece of homology at the end called the pseudoautosomal region. And that small piece of homology creates a weak spot in meiosis for spermatocytes, because if you were to, again, distribute breaks randomly across the mouse genome, the pseudoautosomal region would usually fail to get a double-strand break. So it turns out that mouse cells have evolved mechanisms to boost double-strand break formation, specifically in the PAR. And how does that work? It works in part through boosting the binding of REC114 and other double-strand break promoting proteins. We don't think that the underlying mechanism of the boost is the same, because in the pseudoautosomal region, it seems to be tied to heterochromatin formation and a repetitive mini-satellite called the MO2 mini-satellite. And budding yeast doesn't have anything like that on the small chromosomes. So we think that the underlying mechanism that leads to the boost and binding of these proteins is different. But there's this theme that the cell solves, different evolutionary lineages have solved the same problem using the same toolbox. And we're very excited to try to see how these things are similar and how they differ in different species. That work that I told you about in mouse is part of a long-standing collaboration with my colleague in developmental biology here, Maria Jason, and that was led by a really talented postdoc in the lab, Laurent Acquaviva. And so with that, I'll close, and I'll thank, again, the people that did the work. The vast majority of what I told you about in yeast was work of this really talented research scientist, Hajime Murakami. He had some help recently from a graduate student in the lab, Peggy Huang. I mentioned very briefly at the end the work of Laurent Acquaviva in collaboration with Maria Jason. And we had input from some important alumni who have moved on to other positions now, Isabel, Jackie, and Megan Van Overbeek. And I did mention some of the work that Drew Thacker had done when he was a student. And I really have to give a really profuse expression of gratitude to MSK's core facilities, because without them, none of this work would have been possible. And in particular, Nick Sochi in the bioinformatics core, and Anya Svial and Neiman Moibula in the integrated genome operation. They've really been terrific at supporting our research over the years, and in particular for this project. And of course, the funding is critical for this. My lab is supported by the Howard Hughes Medical Institute, and the work that I talked about today, Hajame's work, was principally supported by the NIGMS. And I'm happy to take any questions.
Okay, thank you, Scott, for a very nice talk. And I think I'm particularly intrigued by the booster sequences. So you talked about the 3 little chromosomes. They behave seemingly quite differently from the rest. And in your translocation experiment, I think you focused on chromosome 1. So are the booster sequences in the other chromosomes similar? Like, do they have different features?
Since we don't know what those features are yet in detail, I can't answer that. So we like the idea that it's the same mechanism on all 3 small chromosomes, but it's conceivable that it's different underlying mechanisms. What I can tell you is that there are chromosome structure proteins that we call axis proteins, HOP1 and RED1 in yeast. And these are critical for this. This was actually first, the aspect of this was first shown by Franz Klein again, showing that the axis proteins are critical for the high level recruitment of these double strand break proteins to the small chromosomes. So we think there's some structural feature of the chromosome that's built by the sequence. The little chromosomes are more GC rich, but meiotic recombination tends to generate GC richness as a side effect of it. So we can't tell whether that GC richness is cause or consequence. So yeah, we just don't know yet.
I see. So have you tried to do similar translocation experiment with, the other 2 chromosomes, or even chromosomes from the other species that are have these small chromosomes?
Yeah, we haven't done that yet. What he did do, what I didn't have a chance to talk about, was a reciprocal experiment. So you can also imagine that if this boost is an evolutionarily constrained feature, that if you made a big chromosome little, it wouldn't have the boost. And sure enough, that's what he found. He made an artificial little chromosome, and now it didn't know what to do properly, and it missegregated at a higher frequency. We're hoping to use experiments like that to tease apart what the cis-acting sequences are. But yeah, at this point, we haven't, we failed from kind of bioinformatic type approaches. So we need to roll up our sleeves, and once we can get back into the lab.
I see. So this question is about the boosted DNA sequences, whether or not they have a higher ratio of cohesing association sites. I think it's, basically the same, along the same lines.
Yeah, so we have looked at a lot of these features to try to get clues, and so far nothing has turned up. So the density of preferred binding sites for cohesin is not higher on the small chromosomes, and I think they might bind a bit more per kilobase, but that's partly because there's a very high level of binding around centromeres, and more of the little chromosomes is pericentromeric, just because they're so small. But if you knock out the meiosis specific cohesin subunit Rec8, it does not eliminate the boost. So the boost is completely independent of Rec8. So we don't think cohesin is the key. We think the other axis components are the key.
I see.
But so again, that just pushes the problem back.
Yeah, I think you got quite a few questions relating to this booster sequence.
I'm happy to talk more about it.
Right. So this question, I actually don't completely understand. So it's asking if the booster sequence is copied multiple times, is the booster effect multiplicative, additive, or it's a purely binary benefit? I think this is actually a great question.
Yeah, so I can answer that for the mouse case, because we actually know what the cis-acting element is in the pseudoautosomal region, or at least we have a very strong reason to believe it's this mini satellite. And Laurent did this very clever, again, a natural experiment taking use of strain and species diversity. There's a subspecies of mice from Japan called Mus musculus molossinus, and that subspecies naturally has a much lower copy number of the MO2 mini-satellite repeat than the B6 strain does. And so he crossed the molossinus mice to B6, and so if the Y chromosome came from B6 and the X chromosome came from molossinus, there was a lot more REC114 bound to the B6 version that has lots of repeats of MO2. And of course, he could do the reciprocal experiment with the parents reversed. And so there, it was very clear the copy number of this mini satellite was correlated with the degree of recruitment of this of REC114 and these other proteins. So because we don't know what the elements are in yeast, we can't do that experiment yet. But that if we can identify the element, that would be really cool to look.
So you're basically suggesting it's maybe additive if it's acting in similar ways.
Well, so if I had to speculate about what I think it is, so in separate work that I, again, didn't have time to talk about, another former postdoc, Corentin Kleist-Boart, who has his own lab in Belgium now, succeeded in purifying SPO11 and all of its partners, including REC114. And he's found something really interesting, and that is that REC114 makes a complex with a protein called Mi4, and the two of them are perfectly well behaved by themselves in solution. They make a heterotrimeric complex that's just monodispersed in solution. But as soon as you mix them with DNA, they make these gigantic condensates. We don't really know quite how to describe them biophysically, still trying to characterize them, but what that represents is very high level of cooperativity for binding of these proteins to DNA. So I think it's quite possible that in yeast, at the early stages of meiosis, when there's very little of these proteins around, the small chromosomes may only need a small advantage in affinity. And because once the proteins start to bind, there's this high level of cooperativity for binding more proteins, I think that even a small advantage for initial binding at low protein concentrations is going to win the race. So that's how I think it actually works. But the problem is that's actually hard to figure out, because it may mean that the little chromosomes only have a very small advantage quantitatively for binding because of this positive feedback from cooperativity. So that's a working idea for how this is going, and we have some of the tools now to try to test it with in vitro recombinant proteins and these strain diversity of sequences to play with.
So we have quite a few questions related to evolution. So I can probably ask together. So it's basically about what's the evolutionary advantage of having these, little chromosomes, and also do these little chromosomes come first and then later acquire this booster sequence, or maybe the booster sequence occur first, right? So like, how does it work?
So these are all really interesting questions. We talk about it in the lab all the time, especially when we're having drinks. So early in the conversation, it makes more sense. Later in the conversation, it makes a little less sense, probably. As for the question of why the Saccharomyces maintains the little chromosomes, I really don't know. So one of the little chromosomes is chromosome 3, which is the chromosome that has the mating type locus on it. And so you can think of chromosome 3 as yeast's sex chromosome. So it has kind of unusual behaviors because of some selective pressures that get put on it for needing to maintain heterozygosity for that if you're a diploid. So chromosome 3 might be constrained by things other than just what I've been talking about. The other two small chromosomes, 1 and 6, they have been conserved for millions of years, and I don't know why. And I really am actually very happy to hear suggestions from people that may have heard this talk, because I really am intrigued by the fact that this seems to be such a successful karyotype that's maintained for so long, but why? I don't know. Okay, so what came first? So you may have noticed when I showed some chip profiles that on average the little chromosomes had the behavior that I was talking about, but within chromosomes there's big differences in different domains. So there are segments of chromosomes. There are segments of big chromosomes that act like they're little chromosomes, and so I think it's quite possible that the boost came first, in the sense that when an ancestral chromosome got bisected to create two smaller chromosomes, it would only be successful if it was already good at competing for REC114. Another way to look at it, though, is that the fitness cost isn't super high from the random model. And so it might not need much of a boost to be reasonably successful early in its evolutionary life. I'm really getting out ahead of myself here, because I don't know I don't know how to model this out in evolutionary terms as to which makes more sense. So I'm just kind of speaking from the gut on this. But I think it's quite possible that the boost came first, or at least that there were sequences predisposed to boosting are the only ones that are going to be successful as small chromosomes at the beginning.
Yeah, so these evolutionary questions are quite intriguing. Actually, there's another one. So this question is asking, so you have this fusion experiment. So the large chromosome now behaves sort of like a short chromosome. Now, do you think, or do you see any disadvantage of this large chromosome thinking that it's a small one?
Yeah, that's a great question. I think yes, because I think break formation is dangerous for the cell to do. And I think the cell has invested quite a bit in controlling so that there's enough breaks, but not more than it needs. And so that's why I've been very intrigued when we discovered, and other workers in the field discovered with us, this negative feedback paradigm for how SPO11 activity is controlled. So basically, the way it works is SPO11's default is to keep making breaks until it's told to stop. And that negative feedback is patterned by success. So when the homologous chromosomes engage one another, the cell stops making breaks on those chromosomes. And so I think the cell really is investing quite a bit of regulatory effort in restraining SPO11. So I think it would, all things equal, prefer not to make too many breaks. So you could imagine that when the Mecate ancestral chromosome 6 fused to chromosome 7, it would be wonderful if we could go back in time and look at the first 1,000 years and see how long it took for the boost to decay. I would expect that the boost probably decayed pretty quickly, because I think that it actually creates a selective disadvantage to make too many breaks.
So this question again relates to this question is probably maybe it's an experiment you've done. So it still relates to fusion of this small chromosome part to the big chromosome. Does the location matter? Meaning where you insert this small booster sequence into, does it affect the consequence?
That's a great question. We don't know the answer, so that's stay tuned, because that's exactly the kind of experiment Hajime wants to do. Make all kinds of different fusions with different proximity to centromeres, different relationships to replication timing, and really tease out how these pathways intersect with one another. And again, getting back to the cis-acting elements, that's also how we are hoping to try to identify what those elements are, and by these kinds of fusion experiments. Yeah, so stay tuned. We're also really interested. Jeff Buca's lab at NYU has made, many of you are undoubtedly aware, has made these really cool artificial chromosomes in yeast, and has removed, basically a lot of the repetitive DNA and a lot of elements, kind of stripped them down to what's necessary for viability. But they have a preprint on bioRxiv with Andreas Hochweigen, I think, has collaborated with them, showing that these artificial chromosomes don't have these behaviors that we're talking about. So they've managed to have chromosomes that are functional for gene expression and viability, but for meiotic chromosome segregation, they've actually lost these special properties. So that would be a really interesting way also to try to get at what these cis-acting elements are, is maybe to reintroduce natural parts of chromosomes into these engineered strains and see what parts are sufficient to give the boost.
So we have another question relating to the small and big. So looking at all the chromosomes, even though the small, they are indeed small, but they seem to be on the sort of spectrum, right? So there's a continuum of from small to big. But in terms of the REC 114 chip profile, it looks like there's a drastic difference for the 3 small chromosomes. So this question is asking about like, there seems to be a threshold here, and what is that threshold, and how is that detected?
Yeah, that's spot on. So that is the way we view the data as well, that there's something really special about the 3 smallest chromosomes, and at least, a major part of it is this boost that I'm talking about. So my favorite idea is it just traces back to that graph I showed at the beginning where Hajime did the random simulation of distributing the breaks, because that's what that looks like too. The 3 smallest chromosomes are the only ones that have any kind of appreciable risk of failing to get a break. All of the rest of chromosomes are just big enough that they're essentially always going to get a break, even if you just left it to chance. So I think there's a threshold effect, given the number of breaks that yeast makes, that those 3 smallest chromosomes are below the threshold or above the risk threshold, so they're at risk. The rest of the chromosomes aren't at risk, so they don't select for it doesn't select for any special behavior on their part other than what's necessary to do pairing and recombination. That's kind of how we think about it.
Wow, that's really nice. I think that's a sign of, when people asking about evolutionary questions, it's a sign of how intrigued they are about all these findings. Thank you, Scott, for a great talk today. We actually, this is all the time we have for the questions. And to the listeners, thank you all for joining us. Please join us again next Monday, May 4th. Our immunologist, Andrea Schantinger, is going to talk about decoding T-cell dysfunction in tumors. Bye, everyone.